Hello everyone. Let's get started. Today we're going to be talking about microbes in the environment. Our objectives for today are first to discuss the kinds of methods that we use when we test certain environments for the presence of microbes. We'll discuss different types of culture media today. We'll talk about why agar, for example, is a good choice as a solid culture media. We'll compare how agar is used in the microbiology lab, in agar plates, agar slants, and what are called agar deeps. We'll compare bacterial growth on and in solid and liquid media so you can see the differences. And finally, we'll talk a little bit about colony morphology and how to assess it on solid media. Remember, microbes are everywhere. They are both on and in our bodies and the bodies of other living things. They are all over the natural environment, and they're present in our man-made environments as well. Most microbes are perfectly harmless or even beneficial to us, but as we've discussed, a few of them are pathogens, and we need to monitor for those pathogens. We routinely monitor several different kinds of environments for the presence of those pathogens. As we've discussed, we have to watch out for our recreational water bodies. We have to check to see what's growing in that water to ensure that it's safe for people to swim. We have to monitor water treatment plants and public water systems. But we also have to check places like our hospitals and medical offices, where microbes sometimes begin to grow in the actual facility and can be a source of infection for patients. We have to monitor our care facilities, including nursing homes, assisted living facilities, and rehabilitation places. And finally, we have to monitor certain industrial spaces because it's critically important that those places stay clean as we describe it. These are places that, for example, manufacture pharmaceutical drugs or delicate technological equipment. When we go out to sample environments like this, there are actually many ways that we can achieve that sampling. We can take samples of surfaces and use that material to inoculate media. We can also take filtered air samples and use the filters to inoculate media. And finally, we can use the liquid that comes from that particular environment, like recreational bodies of water or water treatment facilities. We can centrifuge the liquid, and we can use what's left to inoculate the media. On this slide, you're looking at some pictures of the methods that people use to gather these kinds of samples. In the picture up at the top, you're looking at a gloved hand holding a commercially available swab. These swabs come in their own test tubes that are sterile. And when you take off the cap, you're also taking the swab with you. This sterile swab can then be used to run across a surface and pick up any microbes that might be present. The cap and swab is then returned to the sterile tube and transported back to the laboratory for analysis. Down here, we can see a person gathering a sample from what looks like a very dirty ventilation grate. And over here, we see another person using a swab to take a circular collection of what looks like a floor drain. Again, there are all kinds of surfaces inside medical facilities and care facilities and even industrial locations that need to be monitored. And we're going to take samples from places where we suspect that microbes might be growing. Environmental samples are usually collected with these commercially available swabs if we're sampling a solid surface. On this slide, you can see a picture of a range of these types of swabs that you can buy. They come in different thicknesses, they come with different lengths, they come in different size containers. It all depends on the needs of the person who's doing the sampling. The key thing is that the container and the swab itself be sterile so that you can collect the sample from the surface wherever you're taking it and then return it to that container and prevent any contamination on the way to the laboratory. You should note that these commercially available swabs have a small amount of liquid in the bottom of the tube. It's a type of culture media referred to as transport media, and it's designed to keep those cells that might be on the swab alive just long enough, essentially, to get it to the laboratory for analysis. Transport media is not a nutrient media. It doesn't encourage the cells to grow or divide. It just keeps them alive until they get to the laboratory. There are several pictures on this slide that show different types of containers that people use and different types of apparatus to take samples from different environments. Let's take a look at each one separately. In the center here, you can see a person who's trying to take a sample of water that looks like it might be stagnant. This is not what I would call a recreational body of water. The person is holding an elongated telescoping pole that has a container on the end of it. When you're selecting a sample from a large body of water like this, you have to make sure that your sample is representative of the whole area. So you can't just take one sample. You have to take several samples from different spots in the body of water. And of course, you would need some kind of a tool like this one to reach the center without having to go into the water yourself. Now, sometimes people actually do go into the water to collect these samples. You can see that in this picture here in the upper left-hand corner. You can see two technicians here in waders, and it looks like they're out in a freshwater area that allows recreational swimming, hence the buoy line behind them. These are the kinds of places that have to be monitored frequently, especially during the summer months, to ensure that there are no microbes growing at dangerous levels in the area where people are swimming. So you can see that they're pouring a sample that they've taken into another plastic container in order to take it to the lab. And again, in a swimming area like this, they would not take one sample. They would travel to select spots within the swimming area and collect several samples. You'd want one sample that was out closer to the buoy line. You'd want samples that were closer in to the shore. You'd want different samples along the length of the swimming area. It's really important that you get enough samples so that you feel like you're getting a representation of the whole area that you're trying to sample. Same thing is going on over here on the right. This person is collecting water, not from a freshwater region, but from a saltwater region. Again, probably a beach where people are allowed to swim. It's a little bit trickier when you're checking saltwater areas because, of course, you have to deal with the tide. But again, you'd want a series of samples specifically from the areas where most people are using the water. And at most beaches, that's right up near the shore. Here's an interesting one down here. We have someone collecting water from a well. You can just see the water shining in the bottom of the well. And of course, the person has attached collection containers to some sort of a rope or a chain to lower them down into the well. In a state like New Hampshire, a lot of people live on properties that are serviced by wells. And it is important periodically for homeowners to hire commercial companies to sample their well water to make sure that there isn't anything problematic growing in the well. Last, over here on the lower left, this is just a sample of the kinds of containers that are frequently used to hold samples that come from bodies of water. These are larger containers because, again, they have to hold enough water to give us a representation of what's being sampled. You'll notice too that the plastic material differs from container to container. That's often depending upon which company you purchase these containers from. But they arrive sterile, and with the correct aseptic technique, the sample can be collected and taken to the laboratory without any further contamination. On this slide, you're looking at a person holding a device that is capable of measuring spores in the air. Now, I'm not talking about bacterial spores here. I'm talking about fungal spores. I put this picture in here because it's important for us as we learn about environmental microbial testing to recognize when testing is done appropriately and when it may not be appropriate. These devices are available to people to use in their homes, and they're also used by commercial companies that you can hire to come into your home and take readings of the air. Now, this device is designed to, again, measure the number of fungal spores that are in the air, so they're already circulating in the home. And it can be quite startling to the homeowner when one of these commercial companies comes in and tells the homeowner that there are tens of thousands of spores floating around in the air in their home. Unfortunately, while there are lots of commercial companies that are absolutely legitimate and take samples and interpret samples correctly, there are also some companies that are less than legitimate and use these devices in order to convince the homeowner to have treatments applied to their home to take care of these fungal spores. And the truth is, those treatments are absolutely not needed in many cases. This device can measure the number of fungal spores, but it cannot tell you what type of fungus the spore comes from. It also doesn't tell you whether or not those spores are dangerous for you at all. We haven't talked about fungus yet. We'll be doing that later this week. But it's important to know that fungal spores are very different from bacterial spores. We've talked about how the bacterial spore is a survival mechanism that bacterial cells have when they find themselves in environments that are hazardous to their survival. The bacillus and the clostridium genera, as well as a lot of bacteria that live in the soil, are able to sporulate when they find themselves in a dry environment without enough water, or in an environment that doesn't have enough nutrients for them to survive. They can go through the sporulation process and essentially enter a state of suspended animation, where they'll stay until the environmental conditions improve, and the spores can then return to the vegetative state. We talked about how pathogenic bacteria create spores that are actually dangerous to humans, because if the spores get into the body, which is warm and wet and dark, the spores can return to the vegetative state inside the human body and create sometimes very serious disease. Fungal spores are very different. Fungal spores are not survival structures, but reproductive structures. Fungal species, certain of them, create these spores as their gametes, or their sex cells, if you will. The male sex cell, the female sex cell come together and create a new fungus. Fungal spores are present all over the environment, both inside and outside our homes. And they are more prevalent in areas that are quite dry and dark. So you can imagine there's a lot of them, for example, in basements of old homes in New Hampshire and the rest of New England. These spores are, generally speaking, not harmful to us, though. They are reproductive structures, and if they were to get in our body, they would not cause us disease. So, if you're working with a reputable company, you'll be told that. But if you're working with a company that's less than reputable, they can use the high numbers that will be recorded on these machines to scare the homeowner into believing that there are lots and lots of dangerous spores floating in the air of their home, requiring some very special remediation that usually costs a lot of money, as you can imagine, when really those spores are of no concern at all. I want to switch gears now and talk a little more specifically about how we monitor for microbial contamination in the environment of healthcare facilities like hospitals and doctors' offices. What you're looking at on this slide is a gloved hand holding what looks like a petri dish with agar in it. In reality, this is a very specialized petri dish called a Rodak plate. Rodak stands for Replicate Organism Detection and Counting. This is a plate. It does have agar in the bottom of it, the way an ordinary agar plate does. But these plates are designed specifically for sampling environments. This person is holding the bottom half of the plate, and they have it turned over so that the agar is actually pointed down towards the surface. These plates are made out of a much softer plastic than what petri dishes are made out of. And what the person can do is gently bend the plate, you can just see it in this image, to cause the agar to sort of pooch out a little bit, and where it can be pressed against a surface. The person gently pushes the agar down against the surface. The agar will essentially stick to any cells that might be present. And then you simply take this plate, turn it right side up again, cover it, and send it to the laboratory. The plate is placed into the incubator for about 24 hours to see if anything grows. Rodak plates are used in a very systematic way so that if anything grows on the plate, those colonies, as they're called, can be counted, applied to a chart, and a decision can be made as to whether or not that facility has good enough disinfection procedures in place. When I use the word colony, I'm talking about one of these white spots here on this plate. Each one of these little white spots is bacterial growth. You're looking at what we call a spread plate. Now, this plate was made in the laboratory. A liquid sample or culture, either one would be fine, was placed on an agar plate surface and evenly spread all the way around the plate. Remember, we looked at the device that we use to do this, referred to as a hockey stick. It takes an aliquot of material, liquid material, and the hockey stick spreads it out evenly over the surface. The plate is then put into an incubator and allowed to grow. The cells that were spread over the surface of the plate will divide while they're in the incubator. And as they divide over and over and over and over again, each individual cell creates its own colony of material. So all a colony is a little mound or a little mass of bacterial cells that all came from the same original cell that was placed on the agar plate. Colonies can have many, many, many thousands and thousands and thousands of cells in them because in a 24-hour period, that's how many times that original bacterial cell will divide until we end up with a mass of cells that we can actually see with our eyes. Now, a spread plate is particularly useful when we want to count colonies. And the reason we would count colonies is because it will give us an estimate of how many cells were in the original sample. More colonies means that there were more cells in that original sample that we spread on the plate. Now, over here to the left, we have another agar plate, but this one is not a spread plate. This plate is called a quadrant streak plate. And this plate is made for a very different purpose. You can see that I've got bacterial growth on this plate, but in a lot of places it just looks like a big smear of growth. In other places on the plate, though, I can see these individual colonies growing. We make quadrant streak plates when we want to evaluate what's called the morphology of a colony. All that word morphology means is body form. We can look at a colony and we can evaluate its appearance and its size. And that can tell us something about what kind of a bacterium or even a fungus produced it. Now, when you're making a quadrant streak plate, like the name suggests, you have to divide the agar plate into quarters in your mind. When we're first learning how to do this, we often actually draw lines on the bottom of the plate in order to produce 4 quadrants. We then take our inoculating loop, sterilized of course, and we dip it into a broth culture to collect some cells. Those cells are placed into the first quadrant just as a smear, just as a streak or a smear, as we would call it. The inoculating loop is then sterilized and we place it onto the plate and draw it through this first amount of cells and streak into the second quadrant, back and forth, back and forth. The loop is then sterilized. We place it onto the plate, drag it through that quadrant, and streak it out again a third time, and again a fourth time. The purpose, again, of making this kind of a plate is to try to produce individual colonies that we can evaluate for morphology. The purpose of making a spread plate, in comparison, is to make a plate that we can use to actually count the colonies and then apply a little mathematics and get an estimate of how many cells were in the original sample. On this slide, you're looking at how to prepare a spread plate over here on the right and how to prepare a very similar plate called a pore plate over here on the left. Now, most bacteria will grow very well for us on the surface of an agar plate. In other words, most bacterial species that we are encountering in the microbiology lab grow very well in room air. But there are some organisms that don't tolerate room air very well because they don't tolerate oxygen very well. Those organisms are better grown on what's called a pore plate, P-O-U-R, instead of a spread plate. Let's take a look at the difference between the two. To make a spread plate, we simply take a sterile agar dish, we place a small volume of our culture or sample in the middle of the plate, and then we spread that material around using a hockey stick. The key thing, again, is to spread the cells in the sample all over the surface of that plate so that every inch of the plate is covered. The plate goes into the incubator for about 24 hours, and then we evaluate it. You can see in the drawing that we have individual colonies growing all over the plate. Those colonies will be counted, and then we'll use that count to mathematically determine an estimate of how many cells were in the original sample. Now, one thing I want to point out to you about colonies is that we refer to them as colony-forming units in these calculations that we perform. We don't call them cells. In other words, when I count colonies on a plate like this, and I go to my equations to estimate how many cells are in the original sample, I won't call them cells in my final calculation. Instead, I'll call them colony-forming units, or CFUs. Now, there's a reason for this. Remember, this sample that I spread all over this plate, ideally, it allowed me to separate each individual cell away from all the other cells and give each cell a place on the plate to start growing and produce a colony. But sometimes 2 cells or even 3 cells might land right on top of each other. And those cells will then create a colony that didn't come from a single cell, but actually from multiple cells. So I cannot say with 100% certainty that each one of these colonies represents a single cell. Instead, I have to say that the colony represents a colony-forming unit. Most of the time, each colony comes from a single cell, but we're going to sort of hedge our bet a little bit and call them colony-forming units instead, as our best estimate of how many cells were in the original sample. Now, take a look over here on the left. This diagram again shows us how to make a pore plate. The biggest difference between a spread plate and a pore plate is how we apply the agar. We're going to take a sample of liquid broth, just like we did for the spread plate, but this time we're going to put it into an empty, sterile petri dish. We're actually going to pour the agar on top of our sample. We're going to let the agar spread out, and then we're going to let it harden. Agar is produced as a liquid, and it solidifies as it cools. So by the time our plate cools off, some of the cells in our original sample will be down on the bottom underneath the agar. Some of them will be actually stuck within the solid agar, and even a couple of them may be growing on the surface. The key is that if we have any cells in our original sample that cannot grow in room air because they have a sensitivity to oxygen, a pore plate will allow them to grow out of the air, either under the agar or in the agar. They will still produce colonies. It's just that the colonies won't be on the surface of the plate.